my dear viewer. I am still your teacher Osbert Nwarinda, still here with you. Now, previously, we were looking at glycolysis and all the stages which are involved in it. Now, in this episode, I would like us to look at the significance of glycolysis. What significance is this glycolysis? Does it have any role? Of course, if I can take us something small, we have seen that within glycolysis we have got very many things there, and in just a quick summary, we saw that we had glucose. Glucose was able to be turned into glucose 6-phosphate, turned into glucose 6-phosphate. It was able to be turned into glucose 6-phosphate, and then we were able to say that this glucose 6-phosphate was isomerized, and then it turned into fructose. 6-phosphate, and then we were able to see that this fructose 6-phosphate was further isomerized. Was further phosphorylated, and it was able to give us fructose 1,6-biphosphate, biphosphate or bisphosphate, and then we were able to see that because this was unstable, we were able to see that this fructose 1,6-bisphosphate, because it was unstable, it was able to break down, and we're able to see that it was able to break down to what? It was able to form, it was able to break down into what we are calling pigal, and then we're also able to see that it also on the other side, it also, of course, we said this one was 3 carbon, and also on the other side it was also broken down into DHAP, and then we were able to see this DHAP isomerized, and on isomerizing it also formed pigal, something like that, and then we're able to see that these 2 pigals, each one of them underwent a series of reactions to form what? To form pyruvate. And this one, each one of these ones underwent a series of reactions to form what? To form pyruvate. Now, this is what we are able to see under glycolysis, of course, in the previous video. For those ones who have not watched the previous video, you go back and first check out in details. Now, we're able to see that during this glycolysis, here we used ATP. There was some usage of ATP, and here we formed ADP. And then also somewhere here, that was isomerization. Isomerism here. We're able to see that somewhere here, we're also able to use ATP to form ADP. But we're able to see that down here, we're able to have ADP pick a phosphate from this reaction and forming ATP. And we're able to see that this reaction, also likewise here, we're able to see that in this reaction, ADP picks a phosphate and it forms ATP, like that. Now, we're also able to say that for these reactions here, during this process here, we're able to see that here, as this pigal is turning into pyruvate, here we have 2 ADP molecules giving us 2 ATP. That means we're able to say that this reaction occurs twice. From pigal to pyruvate, we have this reaction occurring twice. Likewise here, we also had 2 giving us 2 ATP. And also we're able to see that during this reaction, during this series of reaction here, we're also able to see that we had our NAD plus, which was oxidized, giving us what we are calling reduced NAD, and that's NADH. Likewise, also on this side, we are also able to see that our NAD, which is oxidized, turns into NADH. That's what we're able to see. Now, when we are here, and we are able to, of course, we saw all these ones were catalyzed by different reactions. Now, when we are talking about the significance of glycolysis in this case, we are asking ourselves, what importance is this reaction? And of course, one of the first importance is that we can all be able to see, that every person can be able to see from what we are having here, just a small summary here, is that glycolysis is able to give us energy, and this energy is stored in form of ATP. So there is one of the significance of glycolysis is production of energy, which is stored in form of ATP. And that energy is what? That energy is, we have said, considering one glucose molecule. If you consider one glucose molecule, this energy is 2 ATP. We have got 2 ATP molecules as the net. as the net ATP which is being produced. Why? Because we said when you check here, we used the 2 ATP as the input. We had to input 2 ATP as an investment, but after on production, here we are, this pGAL gave us 2 ATP. Likewise, even this pGAL from this side has also given us 2 ATPs. That means in total, those are 4 ATP molecules. Now, the 4 ATP molecules, we have removed the 2 ATPs that we incurred and remain with a net of 2 ATP. So it means through glycolysis, we are able to produce ATP. That is very important. That's the first importance. Now, the next one, we are saying from here, we are able to see that the glycolysis gives us something very important, and that is what it also produces, is production of what? Of NAD, which is reduced NAD, or what we can also just simply say NADH. Now we are saying this NADH is very important. Why? Because as we progress, we are going to find out that the next step we are going to have is going to be called Krebs cycle, and then the other step is going to be called electron transport system, or electron transport chain. Now we shall see that whenever this NAD is produced, this NAD will carry this hydrogen, and within that hydrogen it also carry along with the electrons. So when it goes to the electron transport chain, the electrons it will be carrying this NAD which is this NADH will now help us to carry electrons, and these electrons within it will be donated to the electron transport chain. And then as those electrons will be donated to the electron transport chain, as they move through that electron transport chain, those electrons will also produce more energy. So what we are trying to say is that yes, glycolysis gives us ATP directly, but indirectly it also contributes to production of other ATP molecules. How? Because we are saying it produces NADH. This NADH is a hydrogen carrier. It carries hydrogen, but in the process of carrying hydrogen, within it also carries electrons, which it will later donate to the electron transport chain in that third stage. And as those electrons that will be donated by the hydrogen, as the electrons which are carried within this hydrogen carrier NAD are going to be passing through the electron transport chain stage. As we shall see, we shall have more ATP produced. So yes, we have ATP produced directly, but we also have another ATP that's going to be produced indirectly from the ability of this glycolysis producing or giving us NADH2. I hope that is clear. And then we also, not NADH2, but we're saying NADH. Excuse me there. Now, another key significance that we can also look at is that there is a way how this thing can also explain. It also has some kind of explanation and evolutionary. It also has an evolutionary significance. Significance. Why? It tries to explain what we call ancient origin. It tries to explain ancient origin. What do I mean here? If you go and try to do some reading, you will find out that I don't know at which year you are watching this video, but we shall realize that it is believed that this world very many years ago, actually how many years, it is believed that we had got bacteria onto this world. We had bacteria from 3.5 billion years ago. If we go into history and so on, it is believed that 3.5 billion years ago we had organisms on this world existing. We had bacteria that were existing on this world. But now it also believed, also from different studies have also shown that the oxygen was able to exist onto this world 2.7 billion years ago. 2.7 billion years ago. So we are asking, if we had organisms like bacteria 3.5 billion years ago, and then we're able to have oxygen start existing onto this world 2.7 billion years ago. That means which one came first? That means we had organs that used to stay on this world before the coming of oxygen. So we're asking ourselves, how were these organisms able to survive when there was no what? When there was no oxygen? So the reason as how they were able to survive is because, I think this one I even talked about in a previous video, they're able to survive because they could be able to produce energy only relying on what? Relying on glycolysis, because we have seen in glycolysis there is nowhere where we have found ourselves using what? Where we have found ourselves using oxygen. That means those organs were able to produce energy only by utilizing glycolysis. So it means this glycolysis was one of those steps that were one of those processes that organisms that we used to live here billion years ago were able to depend on for their survival without necessarily needing oxygen, because oxygen was absent. You see? We are also saying that glycolysis is also very important because it is the basis. It is like the foundation. It is like the foundation of aerobic respiration. It's the foundation of aerobic respiration. Yeah. Even when oxygen is present, you see, we are saying that this one does not require oxygen, but we are saying even when oxygen becomes available, we cannot have any other stage take place without this one first taking place. So all those we are going to find out those other things that will be used, all other materials, all other molecules that will be used in when oxygen is present. For example, those materials that will be used actually will be the materials which would have come from glycolysis. So glycolysis, yes, it doesn't require oxygen, but we are saying that all the other processes that will come later, even oxygen is present, all of those processes use materials, use molecules which come from glycolysis. So it is a foundation for aerobic respiration, as we are going to see in our subsequent videos. Just keep a watch. And then we are saying that also this glycolysis also tries to explain, or it also plays what we call a survival role. It plays what we call a survival role. How do we explain this one here? This glycolysis tries to, and it enables organisms to survive. For example, during anaerobic conditions. During anaerobic conditions, you can be there and you find yourself in a situation where oxygen is not enough. You can be here when you are anaerobic. For example, me and you, we are aerobic organisms. We get energy after we're using oxygen. But we are saying under some circumstances, or under some, as we are going to see in the subsequent videos, you may find yourself being in a situation where there is where oxygen available is not enough. So the question is, how is your body able to survive? How is your body able to enable you to survive? We are saying the body is able to carry out this glycolysis. Even when oxygen is not enough, the body is able to carry out glycolysis and give you energy without necessarily, even when oxygen is not enough. Why? Because we are going to realize that for other stages that will come, the other stages which are going to come later, there is a way they require oxygen to first be present, and therefore when oxygen is not enough, then it means those stages might be hindered in one way or the other. But we are saying even when we don't have enough oxygen, even when we have found ourselves in a situation where there is no enough oxygen, we are saying glycolysis is so important to us because it will enable us to continue having some energy that can enable us to progress as we wait for the oxygen to become available, and very many other things, of course. So these are some of the things. Of course, I'm not exhausting them. You can search for even more other importances or significances of glycolysis, but these are just some of a few things that I can share with you for now. So we are saying that glycolysis as a stage is so paramount, as a first stage of respiration is so paramount in these different ways. Now, in our next video, we are going to expound on this. We are asking ourselves that yes, we have seen that the glycolysis leads to production of pyruvate. Now, we are asking ourselves, so After pyruvate has been formed, what happens next? After you have formed the pyruvate, what next? So that's what we are going to address in our next video. Please follow us. Thank you very much. May God bless you.